Yeah, so I was going to talk about the stuff we're doing in periodically flooded soils, but as my esteemed chair is also working in that area, I figured we would try and talk about something else. And Eddie gave me a mandate to keep people entertained and wake people up early in the morning. So here I am waking you up. So I'm going to have some fun. And fun for me involves essentially exploring many different research areas, anything to do with microbes and microbial genomics, and trying to explore how we can intersect across different scales of investigation, both temporal and spatial, and really start to understand the mechanistic metabolic elements that underlie microbial ecosystem dynamics, right? So we, about 5 years ago, started up the Earth Microbiome Project, which is a multidisciplinary, multi-collaborative effort to try and catalog the microbial diversity of the planet. And using that information, we've been able to grab samples from various collaborators across the planet and start to piece those samples together into a strategy for examining microbial dynamics. So I'm going to start at the level of the planet, or at least at the continental level, right? In these kind of investigations, we've been examining how bacterial communities show biogeographic distribution and what influences that biogeographic distribution. And as part of that program, we've really been starting to develop an understanding of the links between high-scale processes such as microbial community structure and fine-scale processes and higher-scale processes such as climate change. So in this video, we're actually seeing a change in the microbial community structure as an average every year since about 1908, going up to about 2011 on the Tibetan Plateau. And this is work from Joshua Ladau's group in collaboration with my group and Katie Pollard and Haiyan Xu from China. And what we're really trying to do here is examine how, at a very, very coarse scale, how community structure is impacted by changes in large-scale processes such as weather patterns and climate and land use. And this is helping us to really start to understand the microbial structure at this very coarse scale. And so what we can do with this is start to understand how that microbial community links into climate change, right? On the basis of that previous model, it looks like the microbial community we find in the soils now is best predicted by the climate that occurred in about the 1970s. So in around 1975, the climate started changing the soil, and it started having an impact upon the plant communities in that soil. And the lag in that change is only really starting to be felt around now. So that premise allows us to potentially extrapolate into the future and stipulate how changes in climate over the next, well, if the microbial community actually equilibrated to the current climate patterns that we see now, what impact that would have upon microbial community structure and richness and function over the next, say, 30 years. So we would see like an increase, actually, in species richness across most of Tibet and North America. But whereas in Tibet, it would actually be quite universal in an increase. In North America, it would actually be very, very patchy increases. So a lot of areas staying quite standard, areas going down, areas going up. We can examine how that's affected at different levels of phylogenetic resolution. So examining, say, alpha proteobacteria and seeing how it would change in Tibet or North America, we actually see an inverse relationship. So biogeographic regional responses are likely to be highly, regional. Easy. So alpha proteobacteria is increasing in Tibet and decreasing in North America, and we know that shows a relationship with carbon-nitrogen ratios. Therefore, we can start to explore the potential implications of changes in plant community structure, changes in edaphic variables, and that microbial community balance. So this is, again, very coarse, very high level, but it gives us the ability to start to focus our research agenda on specific areas. The reason we got into wetlands and these flooded plains is because the microbial community structures in those were really hard to predict because of that stochastic variability. And so these kinds of very coarse scale modeling efforts, while in and of themselves aren't very helpful in helping us to understand what's going on in a single field or in a single lake or river, they are very useful in helping us to pinpoint the areas we should be investigating more. And so the work we've been doing with Chris Henry, and work Chris Henry's been doing with a lot of people in this room, in fact, over at Argonne National Laboratory, is to go to the exact opposite scale of that level of complexity and start to examine the metabolic relationships which underpin some of those dynamic biogeographic temporal structures that we see in the microbial community. So, Chris takes the metagenomic data that we generate in such high levels and the JGI generates in such high levels and starts to take that and assemble and bin it. We do a lot of the assembly and binning into particular genomic structures, and then he takes that information and starts to predict the metabolic structure, the metabolic capability of each of those organisms. And he, using him and his colleagues and KBase doing this initiative has started to build these metabolic models, so metabolic flux balance models for 5,000 or 6,000 individual bacterial taxa at this time. And we're leveraging this to really start to describe mechanistically the underpinning of the biogeographic and temporal dynamics we see in that community structure. And one way we have to change what Chris is doing in order to manipulate this is to take his metabolic models and link them together. So an individual organism's metabolic flux response to an environmental stress or environmental change elicits a biomass and a biomass change for that organism and a particular metabolic interaction with its environment. So it will either consume metabolites or generate metabolites. And those metabolites can be taken up by other organisms. And that metabolic relationship between one organism and another organism and between organisms and their environment is the underpinning of the change in the biomass of the whole community, and also the response of that community to any host it has to deal with in its environment, be that host a plant, an animal, a human, or even other organisms, other microorganisms in the system that we're not necessarily modeling, like fungi or protists. And this is highly important. Each one of these organisms has its own metabolic reactions and its own metabolic relationship. But if we can learn how to intersect them, it becomes a more important, more interactive element. And there are lots of individual groups around the country, around the world, doing this kind of analysis right now. It's just at a very nascent stage of being able to translate this into an ecosystem dynamic option. Some of the work that's been going on with Dave Westin and Chris and Sam Seaver at Argonne have been trying to translate this information, is to take the cyanobacterial communities that interact with sphagnum moss and basically analyze the metabolic relationships of these systems, right? So the cyanobacteria are nitrogen fixing, and they take in that nitrogen from the environment, and they translate it into ammonia as a metabolic output, which is then taken up by the sphagnum, okay? So you see that dynamic relationship in the system. This is actually a host-microbe relationship, and one that Chris and his team, along with Dave's data, can actually model and predict with a reasonably high level of accuracy in terms of the change in biomass of the individual organisms. So this is a success story in my mind. And we're working with Chris, and there isn't pretty much a week goes by when we don't throw him another metagenomic data set to try and help say, look, this one would be really cool. If we gave you all of this data, could you create a model to help us predict how this thing would change, how it would alter its metabolite environment? And we can, we use metabolomics to analyze the metabolome of a particular system, use transcriptomics and proteomics to determine how the different functional genes are being turned on and off. And then we use that information and give it to him and say, look, can you take all that data and predict how this system responds based on our observations? And it's getting better. Obviously, a lot of the metabolic relationships embedded in these cells can't actually be annotated at the moment, so we don't know what those genes are doing. But I've never ever been held back by someone saying it's not possible. It is possible. We just crap at it at the moment, and we're going to get better. But that's the key point. We will get better, and we will get have a much greater ability to annotate and understand how those functional responses are working. And so if you look at any of the new work that's going on in this area, cheminformatics, for example, is a great example of this. Being able to predict the gaps in your metabolic model and relate those to the environment, I think, is very important. So we've been working with this kind of information, but looking at it in a much more complex environment. So work we've been doing with Janet Jansen, now up at PNNL, and Kim Handley in my group, has been taking metagenomes and metatranscriptomes generated in part by JGI and our lab from the Gulf of Mexico oil spill through the BP funding stream, and starting to understand how those metabolic relationships in that community are manifest when the community is exposed to oil. And we see all the same kind of relationships. Like the Beggiatoa are doing some nitrogen transformations, and then we see some of these delta bacteria and the Colwellias and the Psychocassicus that everyone else predicts. And we can basically take the metagenomes, assemble those genomes very easily from this very complex environment. Well, I say very easily. It seems very easy to me, but I'm sure Kim rips her hair out on a regular basis trying to work through this. And then take that information and predict what these big players are doing. And in doing that, we noticed gaps in how those organisms were processing all of this wonderfully new hydrocarbon mixture. And what they were doing was they were taking all this new hydrocarbon, and then you get this weird hydrocarbon, you get a nitrogen deficit because you've got all this lovely new hydrocarbon material. There is nitrogen moieties associated with those hydrocarbons, but these organisms start to go and look for new recalcitrant, previously recalcitrant, nitrogen forms in the sediment in order to fuel their biomass growth. And we see a very similar thing when plants invade another plant system. They manipulate the below-ground microbial community to actually, by pumping in lots of carbon compounds, and then those microbes that can utilize those new carbon compounds often utilize new recalcitrant, previously recalcitrant nitrogen sources. So what we see here is that these organisms seem to change the environment, and then these other organisms, the gamma proteobacterial groups that underlie those communities, have a much bigger impact on changing the nitrogen turnover ratio of that environment, right? So these gamma proteobacteria are aligning with particular hydrocarbon niches in this complex hydrocarbon mix, and they are then activating new nitrogen forms in the sediment. And therefore, you get this sudden release of bioavailable nitrogen into the sediment, which fuels this massive burst of biomass productivity. This is helping us to understand the ecological successional dynamics of this system, which for me is the most interesting point. We've disturbed it, and yet somehow it's flourishing. And that flourishing component, that biomass production, I find to be the most interesting driver of our understanding. But we've got to go back into the metabolic model of each of these organisms and identify those flux characteristics. So the human body is just like, well, my human body is, just like sediment from the Gulf of Mexico or a plant system or the continental United States. It's just another environment with lots of ecosystems on it. And those ecosystems are just as interesting as any other environment in the world, especially when there are funding agencies out there that are willing to give you a lot of money to look at it. And there are lots of overselling of the microbiome. That's one thing we have to be aware of. We can't cure everything with the microbiome, but there's possible ways to use that data that we get to actually transform medicine, and that's what we're mostly interested in. So we know that bacteria can protect against food allergies. So work from Dion Antonopoulos at Argonne and groups at Chicago have shown that adding Clostridia back into a mouse model that has a peanut allergy can eradicate the response to the peanut stimulus. So essentially, the Clostridia appears to dampen down the immune activation response that the peanut activates. And that's very interesting to us. And we've been digging down into this with milk allergies in human beings and various other food allergies. And we've identified particular trait responses of very specific strains of certain Lactobacillus, so things like Blautia strains and Rosaburia strains that have very specific responses, mostly mediated by small chain fatty acids, in the gut response that feeds back upon the immune system. This is very important in our understanding of how the microbes then mediate their environment. We've been exploring through American Gut, which is a program that Rob Knight and I and a few other people founded a couple of years ago, where we get the public to crowdsource a funding program. we've been looking at allergenic children in the American Gut Program. And this kid here, the little hexagon, is that hexagon? Little hexagon there that says sun. That hexagon that says sun, that person is highly allergic to virtually everything. This is one of those kids you get on the bus where if you open a jar of peanut butter, he's going to die, or if you open a banana peel, he's going to die. It's the one that annoys me when I want to give my kids a peanut butter and jelly sandwich. I'm turning American. I'm giving my kids peanut butter and jelly sandwiches. It's just bloody awful. But that relationship, that kid has severe allergies. And by examining, by taking a metagenome from each one of their parents every day for a week, and his sister, we were able to reconstruct the organisms that were most highly abundant and changing and significantly different between those partners. So within this family, we do metagenomes of every person in the family every day for a week. We reassemble those genomes, and then we look at the particular strain level differentiation between the kid who has severe allergies and the rest of the family. And in doing that, we're able to identify particular organisms which may be targets for probiotics or for probiotic development that we could add back to this kid and maybe train his immune system a little bit better. Using this kind of information, we just started up a program with the Shed Aquarium where we're doing exactly the same thing but with dolphins. And dolphins are great because they live in this aqueous medium. So it's not like that we don't have it. We know where all of the microbes that come in contact with those dolphins come from. They come from their handlers, the water, the food, or the air they breathe. And so we have this very easy system that we can understand how each of the microbes interacts with that group. Human beings are really hard to manipulate. They just don't do what you tell them. They have minds of their own, and it's very frustrating. But we, theoretically, using animal models, can take the data we gather from these studies and really start to extrapolate our understanding of how those communities respond. So doing this in an Italian cohort, we took saliva. This is a nice little sideline, but I like this. We took saliva from obese Italians and from lean Italians, men only, and we put it in a jar and stuck a mass spec over the top of the jar and added some wine and slooshed it around a bit and measured the amount of hydrocarbons, the amount of aromatic carbons that were released from that mix. Obese saliva generates less aromatic carbons. By taking the genomes of the organisms in the obese saliva and the lean saliva, we could identify the aromatic hydrocarbon pathways by metagenomic reassembly of those organisms which were responsible for that release. The basic premise is these organisms are responding in a way which overstimulates a lean person, causing them to satiate more rapidly to a food input, whereas an obese person doesn't satiate as rapidly and they drink more. Why we chose wine and not, I don't know, like some food stuff? Well, that's I like wine. Wine's good, right? But this is very interesting, enabling us to pinpoint the exact metabolic mechanism by which the organisms are mediating a response with their host. We know that microbes can make you fat. Everyone knows that. One of my colleagues, Liping Zhao, who was a very morbidly obese guy, was 385 pounds. He lost a lot of weight, around 113 pounds, in about 9 months, just by eating a traditional Chinese whole grain diet. And he found one bacterium, this Enterobacter cloaceae B29, that was really abundant at the beginning and absent at the end. And he put it into a mouse, and the mouse got fat, right? So we know that you've got that one organism which can make a mouse fat. Ruth Lay's work has shown that obese twins, if you take the stool from an obese twin, Y twins, monozygotic twins have a slightly but significantly more similar microbiome than dizygotic twins, right? So if you have the same genome, you have a slightly more propensity towards developing the same microbial community. But still, you can get a fat twin and a thin twin even if they're monozygotic. So the obese twin, you take the obese twin's stool microbiota and you put it into a mouse, and the mouse gets chubby, yeah? Chubby? Is that an American thing? Can I say chubby? Or obese. We'll go with obese. Chubby might be offensive, I don't know. If we take the obese stool and we add Christianicella minuta, the mice stays slim. So with Liping Zhao, we got one bug that makes you fat, and with Ruth Lay, we got one bug that makes you skinny. And you can see the potential probiotic elements. But what's interesting is we still don't know why. And we have very little understanding of how Christianicella minuta works. And so merging the stuff we're doing in environmental science and genomic modeling and metabolic processes into and applying it more readily to this very rapidly growing and uncontrolled field of medical microbiome research will be incredibly valuable. I'll just briefly say this. I'm a chronically jet-lagged person, and I know that when my circadian rhythms in my brain are out, I'm more likely to put on weight than if I'm in a standard, stable circadian rhythm cycle. That's been known for a while. We showed with work in my group at University of Chicago with Gene Chang that if we added high fat to a mouse diet, this milk fats, we could actually disturb the circadian rhythms of the bacteria in the gut without disturbing the circadian rhythms in the brain. And those bacteria, when we generated a metabolic model, release certain metabolites which actually disturb the circadian rhythm of genes in the liver. And the liver breaks down enzymes in the body, and it also breaks down hormones, which are particular enzyme components. And what we actually start to see is when those circadian rhythms in the liver are disrupted, we actually see the mouse gain weight. So the basic premise is that you can alter the microbiome by altering the diet, and it will alter the host phenotype. And this has implications for phytohormones in plants as well. We actually see that bacteria in roots actually show a circadian rhythm. And those circadian rhythms can actually be disrupted and actually affect the circadian rhythms of hormone transference in the plant. So everything's related. This is what I love about microbiology. If I find something in one place, I can find it and I can link it all together in other places. And that's why you can work in every field. Where do the microbes come from? I'm very interested in this. Maria Gloria Dominguez-Bello showed that bacteria that are associated with the vaginal cavity contaminate's bad. They actively colonize you if you're born via vaginal delivery, and if you're born via cesarean, you look like mother's skin. And then you get bacteria from the mother's breast milk. The mother actively recruits bacteria from her gut and from her oral cavity, which goes in via the lymph system and is deposited into breast tissue, creating a symbiotic. It's a prebiotic with lots of lovely fats and sugars and proteins, and then a probiotic of these microbes that are pumped directly into baby. So that's important, right? What do those microbial startup cultures actually do to the child? So a baby that's born vaginally and given breast milk will have this evolutionary predetermined microbiome, right? But it doesn't necessarily mean the kid's going to get fine. We've been working with the Hutterites and the Amish in various places around the US. The Amish live on these small little farms where the one family has one barn and one set of animals, and they all muck in from birth to adulthood. The Hutterites live on big communal farms, and only boys over the age of 14 are allowed in the farm, right? The Amish have no asthma in their population. The Hutterites have elevated levels of asthma over the US average. All of them are vaginally delivered. All of them get breast milk, right? So this isn't the only. There are people out there, Marty Blaze is one of them, that will tell you that everything, if this gets screwed up, everything's screwed up. But it's not the case. This is not the only point in which things get screwed up. It also gets screwed up when you get out into the big wide world and you're exposed to this microbial universe that we live in. And that ecosystem of interaction that we survive and thrive in is the things which colonize our body. And the same things colonize animals and plants in equal measure. And we want to understand that colonization event, just the same as we would with island biogeography. New island pops up, the microbes colonize it. Well, in human beings, our new ecosystem is the built environment. This room in of itself is our microbial ecosystem. It's mostly just populated by our microbes, though. And we are only just starting to understand the impact that simplified colonization event can have on our body's microbiome. So each one of you is now becoming 0.00001% more microbiologically similar every hour you spend together in this room. And the closer you are together, so George and Hal Peter are getting really cozy there, and they're sharing lots of bacteria right now, and so we'll see a stronger interaction. And the microbes on your seats, these pants you're wearing are like loose fishnetting to the bacteria. So everything that's in here is getting deposited onto your seat right now. I was telling someone earlier, we did a study in one of my classes where we swabbed all the seats before and after the class had been there. After the class had been there, I could tell exactly which gender of a person had been sitting in which seat. The vaginal microbiota was very prominent in the girls' seats, and the fecal microbiota was prominent in the boys' seats. And you could tell it. This is no barrier. So that's really important, right? So we wanted to look at how that impacted stuff, and I'm winding up because I'm not sure about time. So the home microbiome study was an effort to try and understand how that microbial interaction we shared with our space was manifest in this system. Pure ecology, but we wanted to understand that relationship. So we looked at families, and if it's red here, we were really good at predicting that the samples on, say, a floor came from that family. So the microbiome of a particular surface was highly predictive, in a lot of cases, of the family that lived there. And that was very interesting to us. Basically, the family moved into a new home, and when they moved into that home, they deposited their microbiome in that space. In fact, one of the families in here lived in a hotel room, and for about 6 or 7 days before they moved into their new house. Their hotel room was microbiologically identical to the new house after 24 hours, right? So the hotel room looked the same as theirs. My hotel room right now looks like my den at home, which my kids and wife don't go into. I am the only one that goes in there. It's a den. There's not much in it. It doesn't even got a TV. It's a bit of a crap den. But it's my space. And so my microbiome deposits in there, and my hotel room now looks like that microbiome. That's very important. We can also forensically identify relationships in that space. So when two people are physically interacting, such as this young couple in blue and red, we can tell that they have a higher microbiological similarity, a higher level of sharing. But just by being in the same space, so this lodger doesn't physically interact with that young couple, we hope. Or maybe here it did. Maybe something weird went on. But it doesn't physically interact necessarily, but it's in the same space, it's sharing the same air, touching the same surfaces, microbiologically becomes more similar. And we can map the microbiological movement of that system. So each one of these edges is a Bayesian codependent relationship, and we can basically overlay taxonomic distribution potential between, say, a hand and a doorknob. This is actually my family, so it's a family of 2 adults and 2 little boys. And we can track the microbial relationships in that space and actually generate a predictive accuracy about how it will turn over if one of those people leaves or if one of those people enters into a space. We can also see what happens when you add dogs into the situation. Dogs act as a microbial supercharged conduit. They basically run around and lick everybody, and then are very promiscuous in their sharing. One of Rob Knight's studies showed that the microbiome of couples living with a dog, their microbiome of the couple was more similar if they owned the dog than couples that didn't own dogs. So if you own a dog, that dog's sharing the microbiome between you two. And this is really important. This study showed that the dogs actually increase the amount of sharing and push those bacteria around more. And this is because dogs are awesome. That's my dog, Captain Bo Diggly. We actually rescued him from Kentucky on the basis of these results because my wife thought, wow, that's cool. Maybe this is something to this. I think she was looking for an excuse to get a dog. But for scientific purposes, this dog is here to help improve the health of my home. I'm interested in putting microbes in your medical records. To do that, I need to develop a sensor, and we've been working on a lamp primer array sensor. You collect a stool specimen here. This is something I got off the internet, but we actually have a prototype where we macerate the stool and urine in the bowl and then take a subsample of that macerated material and pump it through a microfluidic system whereby we use lamp primers to isothermally amplify particular microbiological target signatures from organisms of interest. Currently, we can only do about 48 organisms, but we're working up to doing about 386, and then potentially thousands as we increase the density of our microarray. But you can imagine sitting on the toilet in the morning, as you do, checking your email or whatever it is you do in the morning. I know that's what I do. Normally, I would look at my Fitbit to see how many steps I need today to beat my friend on Fitbit. I've done 6,150. This is why I'm walking backwards and forwards to increase my number of steps. But if I did that, then my app might be able to tell me what I should eat for breakfast in order to be able to stimulate a microbial community that was more in balance with the rest of my ecosystem. I really do like to blur the lines between hard science and bullshit sometimes, but I think that's really important that we can potentially overcome some of those boundaries and maybe lead to a more translational impact for some of this research. Right? This is work from Eric Alm and Lawrence David. It's my last slide. But here Eric up at the top and Lawrence at the bottom did their microbiome, sequenced their microbiome daily from their stool for about a year. You can, the size of the bars is the density per gram of feces. And you can see that Eric on the top is different. He has more Bacteroidetes, and Lawrence has more Firmicutes, more Pteronocoutes, and Actinobacteria. And you can also see events, such as here, Eric ate a dodgy burrito and got salmonella poisoning. And down here, Lawrence went and lived in Bangkok for about 6 weeks, and it changed his microbiome. So using this kind of apparatus, we could potentially regularly check that process. And we're developing the same sensors for working in rivers and also in fields. So we can check the microbiome and potentially the metabolome using sniffing MSMS technology. If anyone wants to talk to me about that, I'm around for the rest of the day. In field sites, so we can understand the kinds of metabolic relationships between plants and microbes. Thank you.
All right, thanks for an outstanding talk. We are pretty close to time, but maybe we can take one question. All right, Asaf.
Is there one?
He's got Asaf over here.
Oh, cool. Hey.
This is interesting talks, like from New York Times, but what's the title? Title was different.
Yeah, so blame Eddie. Where is he? He's sitting over there somewhere. He's hiding now. So,
because metabolic flux, it seems complicated for metagenomes. And
it's not, but the thing is, the point is, you can assemble a genome from a metagenome, and as long as you've got that genome assembled, you can start to predict a metabolic relationship between within that cell. So you can say, I have this genome, I can predict a metabolic flux model for that organism, and you can potentially predict its turnover of biomass or productivity within a particular system based on qPCR measurements of its actual abundance in that system over time. So as long as you have a validatory measure of its response to an environmental stimulus, you can take the metabolic relationship of an organism you assembled from a metagenome in a particular system and generate a predicted metabolic flux model from it. So we've done that with electrochemical array systems, and electromicrobiologically chemical array systems. So bacteria are growing on cathodes, and it's been highly productive. But when you get into more complex environments, such as the sediment with the oil spill stuff, the relationships are much less likely to be accurately predicted. But there are some organisms where we're much better at it, and some organisms where it's not as good.
All right, well, let's thank Jack for a great kickoff to the morning.